Hi, this is Mr. Waite, and we're going to be looking today at the Makeup Saturday study session for biochemistry. We're going to look specifically at the free response questions. Hopefully up to this point you've already done the multiple choice questions with us. They look like this, and you also did the math grid-in questions. I'd like to focus now on the free response questions. If you're doing the post-play it podcast, make sure that I'm going to go ahead and go over the questions, talk about them really quickly. Then I'd like you to write something in the free response section. Make sure it's thoughtful, make sure it's complete. I would say you don't have to write complete sentences per se, like you would if it were the actual AP test or the mock exam or any of my exams. Since this is review, you are welcome to outline this time, but this time only, and again, that's only for the purposes of reviewing. Just make sure you're not staying in that habit, especially when it's time for the test. So one thing I like to do with my students is when we get to the test, excuse the bell there, the first thing we're looking at is look over all of your questions. It's a great practice to get into because some of the questions are going to be much easier than the others. So this is the first one. It's about proteins and describing the structure of proteins here. We could look at some other ones. This is number one. Number two is about water and asking if you know some things about water. So maybe if you're more familiar with water, you want to start here. And the third one is asking you to analyze some graphs about enzyme activity and respond to those. The thing you should notice on this one is it says it's a controlled experiment, and those should bring a smile to your face. They're not easier, but they're not easy, but they're easier than most questions. We'll talk about why here in just a minute. So let's go back up to that first free response question. That's asking about proteins. I would just ask right now, if you're following along in the podcast, go ahead and I'm going to pause it and let you respond to this. Respond thoughtfully, respond thoroughly. It doesn't have to be extremely long, but do your best, and then we'll go over the rubric for this. All right, so let's look at the rubric for this first question. One thing I forgot to do is talk about how I like to identify questions. In fact, let's just do that next time. For this question, let's go ahead and get started on the rubric. So it was asking you about either the chemical composition of proteins, or the levels and structure of proteins. So let's see what the rubric says about both of those. Really quickly about the chemical composition, keep in mind amino acids make proteins. I didn't mean to cross that out, we just want to highlight that. Amino acids are the building blocks of proteins. They're connected by the peptide bonds. The other thing you could talk about for some extra points is to talk about how the amino acids themselves contain an amino group, carboxyl group, and an R group. And last thing, you could have talked about what proteins are made of: carbon, hydrogen, oxygen, and nitrogen. 2-point maximum. Let's move on to the next one, though, which is the levels of structure. And a lot of students sometimes struggle with this. But we're looking at the primary structure, first of all, which is just the amino acids lined up, connected by their peptide bonds. So I do think it's important just to recognize if you stand back and look at them, it kind of looks like just a string. Secondary structure of these guys, they have a helix or pleated sheet shape, and they're being held together by hydrogen bonds. I like to think of it as the string of amino acids being folded over. And again, this is where the hydrogen bond interaction would happen or take place in here. Next one is the tertiary structure. This is a result of the R groups on the amino acid, and if I were to draw it, I would look something like, okay, we have this thing, it's folded over. Now if we take that and twist it or fold it over again, we start to get a three-dimensional structure to this, and that's where the tertiary comes into play. And then lastly, we get a quaternary structure. And the way I tell my students to think about that is to think about two tertiary structures interacting or coming into play here, and this entire thing would be my quaternary structure. This says two or more polypeptides are interacting. And again, look at all the different types of bonds that participate in that interaction. Here's a great picture to show you these at a little bit better level. So we have our primary structure, amino acids connected by peptide bonds. Here's a secondary structure, like taking a string and wrapping it around your finger. Tertiary structure is taking that string off your finger and wrapping it around itself. And then finally, the quaternary structure is two or more polypeptides being bound together. All right, let's go to our next question, and this time I want to give you a better idea here for what we're looking at. Did I skip the next question? Number 2. Let's see, there's number 1. I must have missed number 2. Dang it. Okay, here's free response question number 2. I'm going to go through and identify this. Actually, let's look at it in the same format that you have on here. So let's just scroll down a little bit. It's talking about the role of water. And again, this is what I like to do with my test, or I teach my students to do this with their FRQs, is to go ahead and circle the command word, which is describe. And what describe means is you can't just list it, you've actually got to talk about it a little bit too. And what are we describing? The properties of water and how they specifically pertain to 2 of the following. I put boxes around the numbers to know I need to answer at least 2 things here. So I could part A here, I could put part B down here, but I want to talk about 2 of these 3 possibilities that they've given me. What are those? The role of water as a medium for metabolic processes. That's the key word there. The other thing that we're looking at, again, metabolic processes, the role of water to moderate temperature. That could be within organisms or the environment. And finally, you could also choose from the movement of water from the roots to the leaves of plants. Choose 2, the 2 that you are the most familiar with. If you write about 3, keep in mind the readers are only going to grade and read the first 2, so it's really not worth your time to write about the 3rd one. Just to choose the 2 that you know best. We're going to pause it right now. Go ahead and write your response, and then let's look at the rubric. All right. So the rubric for question number 2 looks like this. Water's role as a medium. You could talk about how it's a solvent and it dissolves a number of things. Don't forget, water is the universal solvent. And so the other thing you could have also talked about is the movement of water in this process of osmosis across membranes. Don't forget, it always goes down the gradient, goes from a high concentration to a low concentration. The next thing you could have talked about would be water's ability to moderate temperature. And a key word here is either specific heat or specific heat capacity. And what the water is doing, water and air is going to either absorb heat and make it cooler if it's near a body of water like the ocean, or it's going to give off heat and make it a little bit warmer during the cool season. Again, you have to be near a large body of water. You could talk about how heat evaporates quickly. The evaporative cooling process, water turning to gas, is going to need energy. If it's stealing energy from the organism, it's going to cool down that organism. You could also talk about ice forming on top of a lake and how that keeps the water underneath into a liquid state and allows aquatic organisms to survive. Last thing here they're looking for are the movement of water in plants. Key word here is transpiration, and it's all driven by the sun through the opening of the stomata. Keep in mind, the leaves go up the roots, up the tree trunk, and out of the leaves. That's only because the leaves have the tiny stomata on the bottom of them that open and allow it to go to, again, a lower pressure difference down the concentration gradient. Last thing we're looking at here is capillary action of water. That's just how it makes its way up. And the 2 things that are responsible for that are cohesion, water attaching to water, or adhesion, water attaching to other objects. Again, that was a 4-point question. Let's take a look at the last question on here. Looks like this on your sheet. There's 2 parts, part A and part B. Let's focus on part A first. And it says to discuss. So I want to make sure I'm talking about it. And what am I talking about? The relationship between structure and function of this enzyme. How does its shape determine its function? And then it also wants me to explain how the structure is affected by temperature and pH. So 2 things it's looking for part A. Part B, describe a controlled experiment. And guys, again, controlled experiments, they're not easy, but they're easier. Make sure you hit all the parts of a controlled experiment to get the full points on this one, and it should be a little bit easier. Part of a controlled experiment is a hypothesis, which they're also telling you to be sure to include. When you're doing this, make sure you recognize that they're looking at two different things here. It's always analyze your axes. The y-axis is the enzyme activity. You can see how it kind of has an optimum temperature right here. This is enzyme activity plotted over pH, an optimum pH about right here. When enzymes don't work, they change their shape and they denature. That the enzyme activity greatly decreases. Other stuff, if we're talking about a controlled experiment, we already mentioned the hypothesis. Don't forget, and hypothesis always have the if blank, then blank. Make it pertain to the topic that we're talking about. Don't forget your independent, dependent, and independent variables. The independent is the one that you're changing, which would be temperature or pH. And to talk about doing the experiment multiple times to get better data, that's always an easy one. I'll let you start writing now, and we'll go ahead and look at the rubrics for this. All right, for part A, this is what we're looking at. Things that you could have talked about, how there is an optimum pH or temperature. You could have talked about how the enzyme substrate fit together like a lock and key. They have to be very specific. You could have talked about the tertiary structure of the enzyme being affected by either the temperatures or pH and then not working, and a description of the enzyme structure or function, and they give you possibilities what those structures could be over here or the function of those over here. We're going to not spend too much time on that so we can keep going with this. Another important word is this idea of denaturation, and that's when the enzyme denatures, it changes its shape, and then its function would change as well. So it kind of breaks down after a certain temperature, or in this case, a certain pH. You could talk about how the temperature affects the conformation of the bonds. And again, basically, the higher temperature is more likely to break certain types of bonds. It's always good to throw out words like hydrogen bond is holding those together and affected by the temperature. You could do the same thing for pH, but when you're describing pH, remember it's a measurement of the hydrogen ion concentration. Those interactions amongst the hydrogen ions would also affect bonds, such as the hydrogen bonds. And then this last one down here, kinetics. Kinetics is talking about movement. Anytime you increase the temperature, you're going to increase the movement of the molecules in there, and that could also allow bonds not to happen and decrease the overall enzyme activity. So that's for part A. Let's take a look at part B right now. Again, these are the ones that I really want my students to do well on. The design and experiment. So look at what they're doing here. Things where you could get points for this is just describing what's being measured. And what were we measuring in this? The enzyme activity based upon either the temperature or the pH. And this is saying, how do you know the enzyme was working? And it's using this, don't forget enzymes, they take the substrate and they turn that into a product. So anytime you can use some scientific language in here, that's extremely helpful. How is it measured? You could do the same thing up here. We know the substrate's producing a product. We could be looking for bubbles like we did during our enzyme lab. This also talks about identifying the variables. Again, I mentioned this, but the independent variables. Independent is like manipulated, things that you changed. Temperature and pH, those always go on the x-axis. Again, these are the independent or manipulated. And then you could also talk about the dependent variables. I'm seeing if that mentions those here, which in this case was the enzyme activity. It says, describe the experiment could produce these data. As long as you give a reasonable design that could produce some type of data like this, what I think you'd be looking at is different testing the enzyme at different temperatures, testing the enzyme at different pHs, but you need to specifically label what those temperatures are or the pHs are, and make sure you go back and look at the graph for that. Do you hold everything else constant? So remember, you're allowed to change one thing. In this case, it goes either the temperature or the pH. If it's a controlled experiment, everything else needs to be held constant. Don't forget to include a control group. So maybe one that does not have an enzyme. Again, all of these are easy points. So make sure when you're asked to design an experiment, first of all, make sure it's a good thing, Because if you just mention there's lots of